I wonder if I might get a little dialogue between Jason and Mark around the relative importance of T cell and B cell interactions and protection against infection.
Thank you, Jason.
I think it's on. Yep.
Oh, okay. It depends on the infection and the route and how that's all acquired. If you take different examples of, for example, one of the most potent examples of antibody-mediated protection may be the HPV vaccine, right? So there you have something that where it's a virus that's very sensitive to neutralizing antibodies, and it's also highly immunogenic and makes a lot of neutralizing antibodies. And then the third factor is that it needs to, at infection, get to the basement membrane. So it gets bathed in serum neutralizing antibodies. So all that biology is working for you in that example. And the worst example, of course, is HIV, the most difficult. The surface glycoprotein is highly glycosylated. You need kind of specialized antibodies to get in there. And that's just to generate autologous neutralizing antibodies. Then you have to think about the incredible diversity of HIV. So it depends there. And it may be that for HIV that the cellular response is going to have to play a better role. And I hope I convinced people, at least in part, that we were actually, in some respects, getting closer. We're getting closer in antibodies. No question that we're now inducing autologous neutralizing antibodies in humans, but we were also getting closer with the cellular response.
Okay. Yeah, for our perspective, as Mark mentioned, it really depends on the pathogen. For structure-based antigen design, we're really just focused on antibody elicitation, trying to make sure we get the conformations right to elicit a certain type of antibody. T cells are obviously important. You need T cell help to mature antibodies. You can do some things like encode T cell epitopes. I mean, the glycoproteins themselves generally have T cell epitopes, so it's not like we're ignoring them completely. I think most vaccines that have been approved, one of the main correlates is elicitation of neutralizing antibodies. I don't know, maybe Shingrix, is that more neutralizing or?
So Shingrix is a very strange vaccine in that it doesn't target the entry apparatus, it targets the immune evasion apparatus. And I thought when I went to GSK, I would learn the secret of how Shingrix works. I still don't know.
Okay, yeah. So there are some exceptions. Yeah, and it's interesting to think about that. And different groups are coming up with ways of maybe stitching together multiple T cell epitopes in a line, or creating T cell chunks that contain multiple epitopes but still have the protein fold. So yeah, certainly important, but it's going to be pathogen specific.
Great. Thank you. I'm going to ask the next question, and this is a question for Claire. Thank you, audience participants, for submitting your questions. How many products have come to market via nonprofit pharma? And how do the for-profit partners for these products make money with these products? So just help us unpack that financial motivations and understand a little bit of what's happening with the development using nonprofit mechanisms.
Yeah, thanks. Good question. I don't know the number off the top of my head, but there's actually some interesting resources from one of your colleagues, Julia Kendall, I think, from GHIAA, that has actually a really powerful documentation of a lot of these nonprofit public-private partnerships for pharmaceutical development. So for whoever asked the question, I would point you in the direction of GHIAA because they do have a good set of resources to look at that question in particular.
He's fine. He's fine. Okay.
That was dramatic. I'm sorry. Go ahead.
That's pretty.
Yeah.
Oh my, okay.
I'm sorry.
No, don't be sorry. Yeah, and I think from the private sector perspective, that's one of the really tough issues, right? Is to create a kind of pathway to market for private companies to benefit, because these are commercial entities. Public companies have boards and shareholders and need to be responsive to that. And so I think some of what the global health ecosystem has tried to do is establish these different mechanisms. Gavi's the most clear example for today's purposes, but there's a whole bunch of other ones. And what I think is really interesting right now in this space is to watch how this global health architecture is changing to create a new kind of path of least resistance to get these products to market. So in the same way, Lisa, that you're talking about decolonizing or indigenizing the sort of global health partnerships domain, I think we're seeing some of that happening in the global health financing ecosystem, where the Africa Development Bank has just launched Africa Pharmaceutical Trust Foundation, which is dedicated to creating incentives for the private sector to enter into these tougher marketplaces. There's also the new Partnership for African Vaccine Manufacturing, which has done similar work to address some of the need for regional manufacturing on the African continent. And Gavi itself actually is moving beyond its work as a procurement financing entity, and is actually just recently committed to $1 billion to invest in the African manufacturing and regional manufacturing for vaccines ecosystem. So I think all of these new efforts are going towards addressing some of the challenges that there are inherently in being a private company in that space.
Great. Phil, we've been unduly focused on viruses this morning, and you, I know, and GSK have an important role in bacterial vaccines as well. It's opening up a very broad area, but I wonder if you would like to compare and contrast and talk about where we are with the development of bacterial vaccines. Yeah.
So we did talk almost exclusively about viral vaccines, but bacterial vaccines are also undergoing a lot of innovation right now. I think certainly in the pneumococcal space, as an example, we've had pneumococcus vaccines for some time, yet there's a need to push to higher and higher levels of valency to cover the total numbers of serotypes out there. We're now talking about going above 30 valent for some pneumococcal vaccines to capture the full range of diversity out there. So there are new technologies that are involved for pathogens where the primary immunity is against sugars instead of against proteins, and it remains a very active area of immunization.
Great.
So next question is to Patricia Ansari. Getting into some of the science again about the work that you presented. What explains the difference in response to the single vaccination without the booster protein between the different groups in trial 4?
Thank you. So none of the responses were actually measured with just a single vaccine. So the DNA vaccines were given as a prime, as 3 injections at 3 different intervals, followed by the boost strategy with either MVA alone or with protein. So the responses were measured 4 weeks after the boost. I hope it's clear. So it wasn't, the responses weren't measured after the prime. It was measured after the boosting. Yeah, thank you.
Mark, and maybe Phil would have a counterpoint. the question is fairly narrow, and how can you explain how mucosal vaccines are delivered? But I wonder if you'd like to enlarge upon the acceptability of mucosal vaccines, what viruses, bacteria they might be particularly suitable for, and where you see that field going. Go ahead, Phil.
So there are some rather good mucosal vaccines out there. Certainly the live attenuated rotavirus vaccines come to mind. There are others where when they're good, they're very, very good, and when they're bad, they're horrid. And that would be influenza, where at its best, live attenuated vaccines can work very, very well. But the problem of trying to optimize not only for antigenic match, manufacturability, but also when the antigen you're changing, like influenza hemagglutinin, is a determinant of how well it replicates, make it very hard to be consistent year to year. And sometimes when you're relying on these approaches, it's a challenge as you get older and you have some baseline immunity. In addition, while some mucosal approaches like intranasal immunization have done quite well for, say, live attenuated vaccines, there have been big problems when people have tried to do it for protein vaccines. Not only do you not get immunity, but I've been involved in a couple trials where you put something up someone's nose and you get a big immune response, and a very high proportion of people get Bell's palsy, because when the facial nerves comes out of that little foramen, it gets inflamed and suddenly your face droops. So it's not always so easy. But in theory, it's great to be addressing the mucosal immune system, and there's some examples where it works quite well.
Yeah, I think that for academics and people who study the immune response, it's a very, very attractive idea. There are practical considerations which you've mentioned, some of which can be mitigated, and I think that we're about to see a real test of the power of mucosal vaccines in this next-gen vaccine development for SARS-CoV-2. So there are about 30 different vaccines that are being developed for SARS-CoV-2 that are given intranasally. I think that DMID at NIH has chosen 3 that are probably the best of those, and at least 2 of those are replicating. So one is the Mount Sinai Peter Palese Newcastle disease virus that looks quite good and quite promising. The other is a codagenics process whereby they attenuate a virus by codon de-optimization. One consideration, and there's a whole long list of the rest of the 30 are non-replicating approaches, which I think are going to be problematic with regard to durability longer term. We'll see how the data play out, but that would be my suspicion. But there is one thing to think about, and it relates to flu mist, and that is in a population, for example, if you're responding to a pandemic, and now people are suddenly developing a lot of mucosal immunity, for example, to SARS-CoV-2, and you started producing your vaccine a year ago, now if you're making an enveloped virus so that your virions have SARS spike on its surface, like NDV, or like the codagenics approach, you now have mucosal immunity, so there's going to be a higher barrier to you infecting people. So some of the other viral approaches that don't have that enveloped approach may have an advantage there. But I think you need to keep your eye on the data over the next several years, how all these play out. It'll be very, very interesting. But the approach has a lot of advantages with regard to stopping spread. There's probably 25 to 30 papers in the literature, every one showing the same thing, where if you take a vaccine, you give it intranasally, high-level protection in very highly susceptible animals. You give it IM, very little protection. It's the vaccine immunologists love it. There's always developmental things that need to be overcome or considered, but that's where I think we are.
So there was some concern that I was being overly general in my question, and there's a specific question about intranasal devices and how in fact you administer vaccines intranasally.
Oh. Well, maybe a little anecdote. It sounds like it'd be much nicer to have something squirted up your nose than have something injected into your muscle. And I think for many adults that is true. Turns out little babies actually don't like things being squirted up their nose very much. And one little cautionary tale was from the 2009 pandemic, where in terms of making vaccine bulk, nothing was better than making the live attenuated vaccine, because you need very little of that bulk per dose, but there weren't enough squirters. And so it actually didn't have a very big role in that pandemic response. And the simplicity of having a needle and syringe, where needles and syringes are being produced all around the world, there are times when it's a practical advantage. But on the other hand, it works.
It may be that we need something along, in terms of the device, something along the lines of platform preparedness and not just pathogen preparedness, so that you would have those devices ready to go. They're very simple. It's basically a 1 ml TB syringe with a little nozzle on the end of it. Yeah, so it's a fairly straightforward thing, but getting 7 billion of those is quite a step. The other thing is that some of these viruses can be lyophilized, and they can be put onto swabs and distributed by mail. It does not take a skilled healthcare professional to deliver them. And importantly, at least in animals, it seems like doing that bypasses a lot of pre-existing immunity. So you're getting right into the cells that way and bypassing mucus layer and IgA and things like that. So that's another attractive delivery. There are 2 mucosal vaccines in China now. I believe they're both non-replicating Ad5. One is, it's nebulized into this thing that looks like a tennis ball can, and then you inhale it. And that's going into millions of people. I haven't seen the resulting data from that. And there's another that's using this similar intranasal spray device that we used. And I saw recently presented some data that it's not been published yet, where they had given that with a Omicron insert just prior to the, I think it was an Omicron insert, just prior to the lockdown lifting in China in healthcare professionals, and saw a 96% protection from infection, not serious disease. So it's fairly impressive. How long that lasts, I think, is going to be something that we have to study. But it was an interesting result.
Great. Thank you. Another question for you, Claire. You touched on this some, but could you say a little bit more about Gates' priorities, maybe MRI and Foundation? I'm sure they're linked. The Gates priorities in vaccine development in low and middle income countries.
Sure, thanks. So I will just focus on the Medical Research Institute. The foundation has a vast set of investments that they have made in developing novel vaccines across the priority areas in the Global Health Division, notably in malaria, tuberculosis, MNCH. For our vaccine portfolio, we are, as you saw, we have partnered with both pharmaceutical companies, GSK being one, and have a terrific collaboration with GSK in our portfolio products. And we also have worked with the other side of the spectrum, the more academic institutions. So, for example, our Shigella vaccine and development is a partnership with an academic institution in very early stages, and that's for a quadrivalent Shigella vaccine. So we've also explored the space of, as you saw on the portfolio slide, prophylactic antibodies for malaria. We have a study ongoing now, a CHIMI study ongoing now, and we have been exploring what other monoclonal antibodies might be most appropriate for us to take on in our specific comparative advantage in the ecosystem. And I think, RSVs is such an interesting space right now for so many reasons, for the scientific reasons, the market reasons, but also, I think one of the lessons in the RSV space right now is that you're going to, competition is a really good thing. Good competition is a really good thing. And there's certainly room for multiple vaccines, but then also other types of products that are solving for the same problem in different populations. So, for example, there's a number of companies and nonprofits that are working on monoclonal antibodies for RSV, and that's another really interesting space to watch, because in these low and middle income settings, you're going to have populations for whom a monoclonal antibody may be more appropriate for a variety of reasons than a vaccine. So these are some of the questions that we ask when we think about taking on new products into our portfolio. You also notice that we work across a diverse array of diseases, and also through also developing different types of products, not just vaccines, not just drugs. But we actually see our ability to perform and succeed as an organization to be linked to that crosstalk between the different products and between the different disease types.
One more to Patricia, and then we'll do the general one. Thank you. So, Patricia, one last question to you, and then we have one general question maybe that everyone on the panel might want to consider responding to. In HIV vaccine trials, asking, is there counseling and treatment for TB as well, screening and counseling and treatment, and is a possible co-infection rate a possible pretrial variable?
Sorry, I didn't get the question.
Sorry, in the HIV vaccine trials, is there screening, counseling, and treatment for TB that is identified? And is possible co-infection rate of TB and HIV a possible pretrial variable to consider?
Thank you. So for the HIV vaccine trials that we have conducted, the phase 1 and 2 vaccine trials, screening for all significant comorbidities is a requirement. So TB being one, hepatitis B. So whether participants are on steroids, because all this could have influence on immunogenicity. So screening does happen for all diseases irrespective, including TB. And for the phase 2B trial, also it was healthy participants who were recruited. So participants are screened for all comorbidities. I don't know if I've answered the
Yep, I think that
question.
answers the
Yeah,
question.
thank you.
Yeah, thank you. All right, so to finish up this panel Q&A session of the morning, you've all talked about great work that you are engaged in now and a little bit of forecasting into the future, but I love this question in asking you to think a little bit more, look a little bit more into the future. What changes in vaccine development would you each like to see? An easy question you can answer in a few sentences, I know, but yeah. Feel free to, we'll start at this end of the panel.
Sure.
Go ahead.
So, one thing that we saw in pediatric vaccine development was moving from individual vaccines to combinations. And as we have more and more vaccines for adults, we used to be kind of flu every season. Now we got flu, COVID, RSV, more to come. We'll start to see combination immunization for adults as well. So I think that's one trend we're likely to see.
Let's see, in our space, interested in pushing more using AI ML tools for antigen design, higher throughput characterization and screening of variant proteins, and then expanding structure-based vaccine design from viruses into the bacterial space, parasites, fungal pathogens, just seeing how much we can do with these tools.
Let me ask Jason if you think the ability now to go from sequence to structure is going to make all your tools that look at these interactions obsolete.
Yeah, there's been big advances with AlphaFold, the new version of AlphaFold 3, which has been teased. It doesn't generally do well on some of the fusion proteins. It's a bit unfair because it's a single amino acid sequence that folds into at least 2 different conformations. And so the program predicts weird hybrids and chimeras of them. I mean, the goal that people have in mind, CEPI and other organizations, though, is to go from novel genome sequence to a predicted alpha-folded structure to AI/ML predicted stabilizing substitutions to mRNA encoding that without ever having to go into the wet lab. So it's sort of like compress all of that. And we're not there yet, but I think it's doable. Maybe within 10 years as all the tools advance. So I think it's an exciting time. We still need some of the AlphaFold 3, like trying to figure out where antibody and antigens bind to each other. Generally, that still needs to be experimentally determined, although it's an area that's trying to improve. To just give a sequence of antibody, sequence of antigen, and have AlphaFold docket, it's not really there yet, but maybe something. So, I mean, it would be great. Like we're happy to, and we embrace all these new tools.
Thank you. Yeah, why don't you pass it to me? Yeah, so thank you. I think in low and middle income countries, we have learned that for COVID, it was really very fast. We have taken like almost 3 decades, and yet we haven't had a vaccine yet for TB, neither for HIV. And maybe I was thinking perhaps the mRNA technology, I think that is now being done for HIV, perhaps could be a probable answer. But then for a low and middle income country, then there's a need for, I think, things like storage and storage of the vaccines. I think that also has to be taken into an account. Yeah, thank you.
Yeah, in a similar vein to what you shared, Patricia, COVID, I think, made us all really question the status quo of how to bring products to market and caused us to really look in the mirror and say, well, why do we do things like that? Why do we pass through XYZ and not ABC? And I guess I would like to see more questioning of that and more pressure testing and asking questions about, why must we produce X over here and not over here, or not make investments in this type of thing, but only in this type of thing? Because I think COVID really forced us to confront a lot of the historical structures that we've had in place. And a lot of those are critical and should not change. But some of them maybe were good times for us to, like you were saying, Jason, sort of open the aperture to you were talking more specifically to how you develop a actual molecule vaccine, but even in the policy space, open the aperture to new ways of doing business, new ways of entering different marketplaces that can put equity at the forefront. And I see that there's a lot of tailwinds moving in that direction, and I would like to continue to see more of that. Thanks.
I think I would like to see 2 things happen. One is I would like to see a better interaction between academics and those in industry. I think that the industry side tends to think that the academic side knows nothing about the practicalities, development, marketing, manufacturing, all these things, which is in part is true. But conversely, there are many companies out there that have very few, if any, immunologists on their staff, and their idea of vaccines is to jam in whatever the protein is. And so I would like to see those 2 worlds get together. They operate in distinct sort of spheres, and they go to different conferences right now. So I think that's a major problem. I really got an education of that in SARS-CoV-2 when we were speaking with developers, and I'd hear some really, to me, kind of, to put it politely, shocking ideas about how the immune system works according to some folks. But the other thing that I would like to see happen, I think that is even more important, is for vaccine-specific immunology to develop more as a discipline, and not just immunology in general, not just virus-specific immunity, but understanding the importance of all these formulations and dose and extended antigen expression and how those all make better vaccines could benefit both academics and industry. I mean, why should industry have a 100-to-1 ratio of vaccines that actually make it, all the way or go to phase 3. And a lot of academics would tell you that those other 99 did not even have a prayer of being developed. And so I would like to see that vaccine-specific immunology develop better. So.